From Microbe TV, this is Immune, episode number 73, recorded on October 30th, 2023. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back, everyone. How are you doing?
It's a crappy weather day today.
It is. It's pouring raining here the whole day.
Pouring rain. It rained all night. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. Same weather here means it's an excellent day to be inside doing a podcast.
Absolutely correct.
Yeah, I think it's the best thing we can do all the time is just do podcasts. Yeah, but I'm biased. Now, the last time, was the last Immune in Ithaca, Cindy? Was that?
No, because I think didn't we record one without you in between there?
I don't know. Maybe it's.
Or was
it could be. It could be.
I can't remember if that was the last one or the one before.
Let me look it up just to see. We'll go to Microbe TV and let's see all these podcasts here. We'll scroll down.
It looks like Ellen Rothenberg was the last one.
Okay.
It was.
So, yeah.
And then the parasitic worms was the one before that.
All right.
That's right. That was the one we had without you.
Yeah.
You missed a good one. It was fun.
I watched it. I watched it in its entirety. Yes, it was very good.
And we did okay without you?
Yeah, you don't need me. You want me to go? I can leave right now. So, actually, I want to be here because I enjoy it, right? I enjoy your company, and you guys are experts in immunology, so I learn a lot. So I'd like to be here. And I'm here. But today, it's Cindy's turn to teach us, right, Cindy?
We'll see how I do. I found this paper and I thought it was the very coolest thing I had seen in a long time. It is bordering on some systems biology and immunology. I usually tend to pick these papers that I'm not sure I understand fully, but I just think it is just the neatest thing I've seen come out in a while. So we wanted to talk about the numbers of immune cells in your body. And we'll come back to how much weight could one lose if you didn't have any immune cells. It's just interesting to think about.
This sounds like a bad idea.
I wouldn't recommend it. But I mean, you think about, we talk about trillions of cells and how many cells and immune cells. And I think I learned a couple of things. There's definitely some limitations of the paper, but I think it's super fun to talk about. So the paper is published very recently in the Proceedings of the National Academy of Science, or as we affectionately call it, PNAS. And the title of the paper is The Total Mass, Number, and Distribution of Immune Cells in the Human Body. The first author is Ron Sender, and it's from several, at least a couple of groups at the Weizmann Institute in Rehovot, Israel, as well as the Edmund and Lily Safra Children's Hospital, Yeshiva Medical Center in Israel, and the Hebrew University Hadassah Medical School in Jerusalem. So how many immune cells do you think there are in your body? Did you cheat and look?
Billions and billions, to paraphrase Carl Sagan.
So there are 37 trillion cells in our body.
Oh, total cells.
Total cells in our body.
But that's an average, right? Because some people are taller.
It is an average.
Yeah.
Some people are shorter, right?
So that's 37 times 10 to the 12th.
Test our math here, huh?
12 zeros. Billion is 9 zeros.
Yeah, so trillion would be 12.
12 zeros. Yeah.
Okay.
And about 1.5 trillion of those are immune cells.
What is the percentage of that roughly? Can we? All right, let me do the calculation. How's that?
20-something%.
So what are there were how many immune cells? 1.8 times
1.5 trillion out of 37 trillion.
Too many zeros. I'm not going to do it. I can't do it. It's less than 1%, right?
Well, no. 1.8 trillion out of 37 trillion?
Yeah.
It's like 0.5%.
It's like No.
No, it's like 5%.
Yeah.
Okay.
1.8 divided by 37.
8 divided by 37. And they're both trillions. So,
Yep.
yeah, it's 5%, right? Okay.
It's about
Now, in
5%.
contrast, how
And
many
it's
brain
amazing
cells
that
do I have?
I don't know. Billions, right? I think there must be fewer brain cells than there are immune cells.
Oh, well,
No,
there are
immune cells are more
86
important.
billion neurons and 85 billion non-neuronal cells, so it's not as much.
I feel like, but I feel like when I've heard estimates of the weight of the brain, it was more than the weight that they're giving for the immune system here.
That is true because the largest organ, solid organ in the body is considered to be the liver. And that was approximately 2-ish pounds.
I thought the skin was the largest organ. Yeah.
I know. People will argue about the solid organ in one place that you can easily take off and measure. I don't know. It's not so easy to skin a person and measure the skin weight, but
skin and liver, that's the order. Skin, liver, lungs.
Yeah.
Wow. I love these kinds of conversations.
I know. It's kind of fun, right?
It's a lot of fun because you don't think about these things very much, right?
Exactly.
Yeah.
And everything is a numbers game.
Yeah.
Amazing.
Yeah.
Yeah, I was actually really struck when I looked at the number of cells in the immune system because, I talk about VDJ recombination and the number of possible T cell receptors and B cell receptors. And I kind of am like, oh, you can make this huge number and you just make so many that it gets to the number and everything's fine. But I guess I hadn't actually thought about what the number was that you make. It was just in my head, plenty. And so now kind of comparing this to those numbers I quote in other lectures is interesting.
Yeah, because only a fraction of the total immune cells are T cells.
Right.
Or B cells, right?
Right.
So
you're gonna tell us what the different subsets are, right?
Yeah, we'll come back to that.
Okay.
One of the things I thought was interesting as I was reading about this is that they reference in this paper the Human Biomolecular Atlas program. So I thought this was interesting, and we'll have to keep our eyes out for it over the next couple of years because they're planning on imaging 200 cell types in over 80 organs to provide a cellular map of a healthy human, like just the whole thing. And I thought that was pretty spectacular that people are doing this. And I guess it's a matter of collecting samples. They were able to do tabula sapiens and, so we have single cell analysis of all the cells, but this is sort of like mapping them in space, which I thought was kind of interesting. Not all immune cells, but, just all cells. So I think eventually we'll have an idea.
Does this have to be done in dead people or can it be done in living people?
Well, are you willing to give up your organs to be cut up and imaged? I think it's probably on donor cadavers to be able to do this.
Well, it would be cool if you could walk into something and they could just image every cell in you, right? I don't think we have that technology.
Yeah.
Right now.
Yeah. We saw a talk. It's a little off topic, but I thought it was pretty fascinating. So Minsu Kib from Rochester came and gave a talk here at Cornell last week, 2 weeks ago. And he showed an image from a movie, and I forget which movie it was. And basically they put a kid in a thing and it went, and it shined light on him. And it was like, here's the diagnosis, lymphoma. And it went, and it said cured. And so, there's science fiction and then there's reality, but what he was showing was he could optogenetically control CD8 T cells attacking tumor cells, which I thought was pretty cool.
That is very cool.
So you could shine light and turn cells on. So you could put optically controlled sensor, basically proteins onto cells and then trigger them activated by just shining light on it. I thought, woo. And they could do it with melanoma, which obviously is on the skin, coming back to the idea of skin, but they could then put probes inside of tumors with little lights that shine in different wavelengths, and you could put kill switch. I just thought the whole thing was pretty cool.
It's very cool.
So there's like people thinking about how to use light to control cells and immune cells, for example. So
yeah, that's super cool. One of the things that I thought was really interesting with this paper is we also, if you look in again, any basic immunology textbook, there's this table that's happens in, say, chapter 1 that tells you, what percent of cells in the blood are lymphocytes and what percent
Right.
of cells in the blood are neutrophils. And they always give this massive range.
Yeah.
Yep.
So like for neutrophils, it's 50 to 70% or something like that, usually in those kind of tables.
Yep.
And, I talked to my students about the fact that, we don't actually have a specific idea. Is like a person with 68% really healthier or less healthy than a person with 51? Like, we don't know. And so I think that there's a lot more fine definition of what is a healthy immune system that could be done. And
Absolutely.
that is really cool in this paper of sort of starting to get using some of our awesome technology to really get a good definition.
Yeah. I think just talking about blood, one of the statistics that I wrote down that was like a cool fact I learned was, so 90% of those cells in our body are in the blood.
Wow. That's a lot.
Yeah. Right.
Really?
That's what it said, because of red blood cells and platelets.
Oh,
They're so small.
And there's so many of them.
We're
But,
considering that immune cells?
No, just total cells. So if we talk about the
Mm.
37 trillion that are in your body, 90% of them
Oh,
are
I
in your
see.
blood.
Oh,
I thought that
wow.
was pretty. I was like, wow, really? That's crazy.
No wonder humans are so stupid,
I could have read
including me, because
I
there's not
could
enough.
have read the number wrong, but
Oh,
maybe it
gosh.
was 90% of immune cells. But I read that 90% of the cells in the body are in the blood and 0.1% are immune cells. So that just comes back to, like your reference set of information that's in the textbook is based on the blood, right?
Mhm.
And that's actually 0.1% of all of the immune cells in your body. So when we do studies on humans and we take blood out and we use that as our assay, I'm guilty of this as well. How representative of that? That's only 0.1 of the immune cells in the body. So yes, there's different distribution of, different ratios of immune cells as well. But the idea that you're sampling something that's only a minuscule set of all of the cells in the body, and you're trying to base your conclusions off of what you find for humans and using just blood, which I thought was
And I think that with the point that you make there is awesome. One of the points I sometimes will talk about is, there are various numbers out there for what fraction of immune cells might live in association with the GI tract or something like that. And the thing that really matters there, and the thing that's relevant to what you were just saying, is it becomes really hard to measure those things and hard to get a number of, I don't particularly want someone to go through my intestine and look in the walls of my intestine and count how many T cells are there. And so how you address these questions becomes really interesting. And that's one other great part about this paper, is that it's sort of getting at thinking about how we address some of those questions.
So how many cells did you say in the body again, Cindy?
37 trillion.
All right. So I found a paper where they say there are 30 trillion cells in the body and 25 trillion red blood cells. Oh my gosh. So that's what you said.
Yeah.
Yep.
A lot.
Yeah. Crazy.
I guess they're important.
I guess we need red blood cells. There's certainly a lot of them. They don't last very long. You have to turn them over all the time.
Yeah.
Right.
Cool.
So one big question is how on earth do you count? As Fran said, she doesn't want someone digging through her intestines trying to figure out cell numbers. So how do they do that? So first things first, like, what are the immune cells? Like, what are the categories? So they say big categories there's lymphoid and myeloid. Okay. And for those of you who are listening and want to hear the density and the number and the size of your favorite subset of T cells, they don't go there. Okay. So lymphoid means they're testing T cells, B cells, NK cells, and plasma cells. Now that it's generalization. There's lots of different subcategories of that. But what's interesting is that they split out plasma cells because they are a type of B cell. So why those uniquely? And the idea there is because, coming back to Brand's point, the assumption so far, and what I've read, is that the GI tract harbors the most immune cells in the body. And we'll
Mhm.
come back to that idea. And the plasma cells are a significant portion of those immune cells in mucosal surfaces, especially the gut. So it's thought that they are probably occupying a different location than, for example, a naive B cell or a memory B cell. So it was important for them to differentiate plasma cell from B cell. Probably
I
just also the data sets that they had did it so they could tell.
and the other thing, because I had not none of this had occurred to me before I read this paper. I also wonder, about differences in weight of a plasma cell, given its size and sort of its intracellular complexity. Because actually, when just looking at the abstract, when it mentioned the differences in weights of monocytes, I kind of had to stare at it for a while and be like, really?
Who weighed
Really?
a monocyte? How much do you weigh?
I mean, I guess they are bigger, but they weigh what? They weigh differently, and so perhaps plasma cells also weigh more.
Perhaps they do. Certainly, macrophages are the sumo wrestlers of the immune system, and we'll come back to that because I thought that was pretty cool.
So speaking of plasma cells, I once heard that they make 2,000 antibodies a minute.
I would not be surprised.
That
It's a
seems
huge
legit.
number.
Numbers. This is all about numbers today.
Yep.
Uh,
So they divided these cells into lymphoid and myeloid. And so within myeloid, we further subdivide into granulocytes and non-granulocytes. So our granulocytic cells are neutrophils. They're the major one. They have the most numerous in the body, but also the ones we forget about and frequently don't talk about, eosinophils and basophils.
Mostly because they're so rare in blood.
They are. They are not so rare in other tissues, as we'll talk about. And then mast cells. So neutrophils, eosinophils, basophils, and mast cells all have granules, so they're granulocytes. And the non-granulocytic myeloid cells are dendritic cells, monocytes, and macrophages. And again, they did monocytes versus macrophages because monocytes are primarily in the blood, macrophages in the tissue, and it allows you to do a little bit of differentiation. One key limitation, I think, from this paper is that macrophages, we've known for eons that they are very different in different tissues and they have unique profiles and whatever. So in like the liver, we have Kupffer cells. In the brain, we have microglial cells. In the bone, we have osteoclasts. So there are specific tissue resident macrophages that have very unique functions. And they're really just grouping, as far as I can tell, anything that looks like a macrophage together for these purposes. So there are some limitations. But so they define what they're going to say as far as a cell type. Then, of course, you have to figure out like, who are you going to use as your reference, right? So as you said, is it a 80-year-old woman or a 10-year-old boy, or, what are you going to choose? So for this, they chose a male that's 20 to 30 years old, 73 kilograms, which I did the math is around 160 pounds, and 176 centimeters, which is approximately 5'8", and quote unquote healthy. They did we'll talk about it for cell density and mass and other calculations. They did extrapolations that were basically just calculations based on some parameters that they put into a formula to figure out how many immune cells are in a child and how many immune cells are in an average woman. But generally, that's their reference. Then you also have to say, okay, so what are the tissues? Because you can't do every tissue in the entire body, and there has to be a limitation to the data sets that they're able to look at. So they define bone marrow, lymphatics, which includes lymph nodes, spleen, tonsil, so all the primary and secondary lymphoid organs. And then blood, as we've already talked about. Then barrier epithelial organs, muscle, adipose tissue, which is also going to become interesting. Other connective tissue, central nervous system, and then extracellular fluids and matrix.
No liver, huh?
Yeah, they didn't put the liver specifically. They just lumped that in with, I guess, other connective tissue. They kind of lump them all together. So the next big thing is, so they've said, okay, what are our immune cells? What is our reference size human? And what are the tissues we're going to look at? But how do they actually do this? So the first thing they did was they did a literature search for histology, looking for the number of cells per gram of tissue using a whole bunch of search terms and looking at all the different papers and finding out all the different information. And basically what they tried to define was a formula. The formula is the number of cells per unit area in the thickness of sample. So these were all histology. And they took the diameter of a cell and they made this formula to calculate the volumetric density of a cell. And then they multiplied that by specific density of the tissue, which is generally around 1 gram per mil, but it's a little bit lower in adipose tissue because they're kind of fatter and less cellular. And they also, so they use this histology literature data and a little bit of flow cytometry data.
And just to point out, Vincent, for the liver, it is listed under internal epithelial.
Okay.
Oh, that's weird.
At least if I'm reading my colors of green correctly.
Okay.
In figure 1.
Yeah, the colors are a little bit hard to see in some of these figures because
I guess kidney would be the same, right?
Probably.
Yes.
Yeah.
It is the same, I think, color green.
Yeah. So they didn't just rely on this literature search for these cell numbers. They also looked at a paper that used something called MIBITOF, which I had to look up, which was multiplexed ion beam imaging by time of flight. Basically, they metal label antibodies and incubate them with a tissue section. And then the tissue section, the ion beam scans across a tissue section and evaporates things. I'm not sure that's the right word. Then it collects it by time of flight. So it can tell you exactly where that antibody was in that tissue section. So you can look up to 40 different parameters and they can isolate and figure out which cells are where. So they were able to use that too. And then they used something that I got a little lost on, but deconvolution-based methylation atlas. So there's different methylation statuses of different cells, and they have sequencing data and methylation data, and they could tell based on the methylation status which cells it was. So they use these 3 methods to integrate the information, and then they validated some things with tabula sapiens data. So their first data that they show is with these literature search for all the cell volume estimates, and they use textbooks. So they determine what is the size of a cell and get the average. And there was a problem already with macrophages because they cause a problem. And that is that they vary really hugely in size depending on the tissue. But they were able to address that a little bit. And the last thing before you actually get into the data is one has to think that when you talk about what I just said, how they collect how they determine which cell types and the tissue types and the reference person and the data that they're using, there has to be a lot of uncertainty in this. So these are estimates based on calculations and data and things from the literature. So are there exactly 1.8 trillion immune cells in an average reference human? Maybe, maybe not, but it's probably within an order of magnitude, and that ain't so bad for a first pass to try and set a reference for us to look at for the future going forward.
So when they say a person of a certain age, I don't understand how they do that if they're looking at the literature for these numbers.
Exactly. So that is one of the big things is the data that they have from all the different papers that they scanned are not going to all have. First of all of them might not have the ages in there because they may be de-identified. They may not have the information on them, or it may be incomplete, or they may have, some females, some males and whatever. And so they do the best they can to make approximations, but it again, it is approximations.
Okay.
So it's not perfect. So we have to keep that with a grain of salt to think about, exactly what can we conclude from this. But I think the generalizations are pretty fun and cool. So, yeah. Do you have questions about that so far? So that's just really the methods. And then there's some figures that I think took me a while to get through, exactly what they meant, because they have 4 or 5 different parameters on the same graph.
Yeah.
So because they had tissue type, location, size, density, organ mass, and you can go and look at this if you want, but I'll tell you the highlights. One is the highest density of immune cells are, surprise, tonsil, thymus, spleen, lymph node, and bone marrow. So I think most of us would have predicted that's where a lot of the cells are going to be.
So the lymphoid organs have the most leukocytes.
Surprise,
I
right?
know.
And there's some interesting things about like the size of the organ versus the density of the cells. So lymph nodes, if you add them all up, are larger in total compared to like the tonsils or the thymus or even the spleen, which I was a little surprised about. And I had a number. Where did I have a number? There's approximately I looked up how many lymph nodes are in the body, and where was that? Lymph nodes. Oh, I lost it. I think I read there's several hundred lymph nodes in the I'll find it later when we get down there. Several hundred lymph nodes per person.
Mhm.
And so if you add them all together in size, it's actually quite a lot.
Yeah, I can imagine that just from thinking about the size of a mouse spleen versus the size of a mouse lymph node and how many lymph nodes there are.
Yeah, I don't know. We only hear about like, I don't know, maybe 10 types of lymph nodes in the mouse. You think, depending on where you inject, where you collect them from. But
right.
I think there are quite a few.
I think there's a lot of them.
There's a lot. And they're often chains, right? So when you get swelling in your lymph nodes, it's usually you probably had this when you were in anatomy, Vincent, that there's a lot of lymph nodes in the body.
Mm-hmm.
So the lowest immune cell density was adipose tissue. But what's really wild is that it's almost the adipose tissue is in total a huge organ mass because a lot of us have a lot of fat. So there's a lot of fat, but not a lot of cells. But then when you try and look at the total number of cells, it's actually quite a few that are in adipose tissue, because even though it's not very many cells in the tissue, there's a lot of the tissue. So in essence, there's a lot of immune cells in adipose tissue, which I thought was interesting. And skeletal muscle too. There's a lot of skeletal muscle, not a lot of immune cells, but the overall contribution is pretty significant.
So I don't view fat as being necessary to have an immune response, right? I don't view it being infected or anything, but I guess it can get infected, right?
It definitely participates in inflammation.
Yeah, it definitely participates in inflammation, and it also, has a lot of endocrine effects that impact immune cells.
I think that also there's also brown adipose tissue and white adipose tissue, and they have different functions. And brown adipose tissue has regulating thermogenesis and things like that also affects immune cell function. So there's definitely connections in that. One of the things I thought was fun was adipose tissue. Adipose tissue can make up about 75% of body mass, but only 2% of the total count of cells in the body. So another like interesting tidbit.
Fat cells are big. Is that the idea?
They're very big, yeah. And all of the immune cells and other cells are squished in between the adipose tissue cells. So, yeah, that was what they showed in the first one. And then, all of your other internal organs, are labeled green, like you said. So there's skin, liver, and kidney and whatever. And they put colon and small intestine in there. And we thought that those would probably be pretty high in immune cells, but they kind of fall right in the middle with those other internal organs. So I thought was interesting. And if you have a favorite cell type, they tell you the density of each cell type within each tissue. And I won't go into all the details there, but, there's a lot of immune cells in the lymph nodes, spleen, tonsil, and thymus, as one would expect. And they did do some extrapolation for some of the numbers, and I can't remember exactly which ones, but some of it they did extrapolate from mouse or from non-human primate data to add to our understanding of those representations, and they're similar.
The one thing that does stand out a little bit with the immune cell density versus tissue is just looking at blood versus other organs. And so you can already start to see that blood looks pretty different than some of the others in terms of which things are most frequently found in blood and which are least frequently found in blood. We always think about the blood as sort of our model for the immune system. And here you can see blood looking really different than some of these other cell types or some of these other tissue types.
Yeah, and neutrophils are the highest, as one would expect, but yeah, it's a distribution. It's definitely lower, for example, than bone marrow. So it's orders of magnitude difference in density. That's not just total numbers, that's density, right?
Yeah.
So the cool picture, if anybody wants to go and find this, you'll see online many write-ups about this already, is their figure 2, which is the distribution of immune cells throughout the body. And basically each cell type is given a color, and then each organ is shown. And then based on the frequency and the number of cells of that category and all the categories that make up that particular organ, you can see. So for example, bone marrow and lymphatic system have many, many, many boxes. So they make up vast numbers of the total cells. So they say if there's 1.8 times 10 to the 12th, so 1.8 trillion cells in the human body. 7 times 10 to the 11th of them are in the lymphatic system, and 7 times 10 to the 11th of them are in the bone marrow. So that's the vast majority of all of the cells. But if you look, what makes up that 7 times 10 to the 11th in each one of those organs is entirely different. So the lymphatic system is mostly lymphocytes. Surprise again. T cells and B cells, right? Whereas the bone marrow it's basically almost all neutrophils, which is not that much of a surprise because the bone marrow is continuously generating neutrophils that are then being exported into the blood, and they circulate around, and then they are cleared, and they are the major antibacterial defense mechanism.
And,
so it's
and you
not
could
that much of a
maybe
surprise.
imagine if this person wasn't quote unquote healthy, that perhaps there would be more neutrophils that would have left the bone marrow, because that's kind of the point.
Yeah, I think it would. Don't you think it would be time though, right? So if you get
absolutely.
a massive bacterial infection, they're all going to leave the bone marrow, but then the bone marrow is going to go into massive hemopoiesis.
True.
And they're going to, regenerate those neutrophils pretty quickly. And so I think you'd have a short window to be able to see that drop and then repopulation. But they didn't look at, what those stages of neutrophil development are, because you're going to have very early precursors and immature neutrophils, and then mature neutrophils that then exit, that then circulate in the blood and leave. And, if you have an ongoing chronic infection, this would also look really different, because your body would be constantly trying to make new neutrophils, but then it would release them before they're fully mature, because they just got to get them out of their basic training and into the war before, they lose to the bacteria. So there's some interesting things where, we can think about how these might change dramatically during different conditions.
Yeah. And the GI tract also really stands out here, because
Absolutely.
I look at this and I'm saying, like, but I thought the number was so high.
Yep.
You read in some places that the number of immune cells in your GI tract is just, so much higher than in other places.
Yep.
And it isn't particularly higher than others. And it's in fact less than in the lung and less than in the skin. And so I think that some of the commercials I see on TV talking about the importance of all the immune cells in the GI tract and how you're going to have these great products that are going to help them. Might need to look at this paper.
The GI tract is pretty diverse, though. It has a lot of different types of cells.
That is true. With a lot of plasma cells. That's where, and that makes sense. And that fits with, the prior descriptions as well. What I found was interesting, if we want to put some percents on that, may make more sense to people than 7 times 10 to the 11th cells, right? So in general, out of your 1.8 trillion immune cells in the body, about 40% of them are in the bone marrow. And about 40% of them are in your lymphatic system, and 2% are in the blood, 2% are in the GI tract, because it's basically the same. So that's kind of interesting. And I already said that, even though 90% of all your quote unquote cells in the body, because so many red blood cells, right, and platelets are in the blood, 90% of your cells are in the blood, only 0.1% of them are immune cells. And overall, the percentage of all the immune cells in your body that you find in the blood is only about 1 to 2%. So again, thinking about when we're sampling, even all of the incredible work that's being done on single cell transcriptomics and things on human blood and under different conditions, it really only represents about 1% of the total immune cells in the body.
And think about how powerful those cells are if they are representing such a small fraction of the cells in your whole body.
Right. And they don't just live there, right? That's the other thing that's not represented in these data. They're constantly circulating. So those cells that are in the blood are going to go into lymph nodes, for example. If it's a T cell or a B cell, they're going to look for a specific antigen. And if they find it, lots of fun things are going to happen. And that's why a lot of lymphocytes are found in lymph nodes, right? Because they get activated there and then they leave and they do their function. They go into tissues. So the blood is really a transport mechanism for these immune cells, like the immune highway, right? To get it from one place to another.
I also find it interesting that dendritic cells aren't not in a lot of places.
No. There are very, very few dendritic cells.
I mean, there's a lot in the lymphatic system, as you would guess. The skin, right?
Yeah.
Makes sense.
Yeah.
But then some places don't have any.
Right.
GI tract has one square has the GI has one square's worth. Interesting.
Yeah, I also think that, is interesting in terms of how we're defining monocyte versus macrophage versus dendritic cell. And which functions we're ascribing to each of those.
And I did not see, unless you saw, I did not see how they were defining it.
No.
Right?
Right.
So it was based on what the literature said, which we all know can be, there's inflammatory monocytes, there's dendritic, this kind of dendritic, DC1, DC2, DC3, and sometimes there are markers that are overlapping on some of these populations. So again, there's not a lot of dendritic cells, but they may have used a very specific definition of that. And there may in fact be that few, or there may be they may have been missed because they were so selective in how they defined a dendritic cell.
Yeah, I would definitely lump monocytes, or say in some cases the monocytes may contain some dendritic cells.
Probably. I would think so. Because you would think there would be a lot at least a reasonable number in the skin, right? Because we have Langerhans cells and other dendritic cells, and they should be in the lymphatic system. So there's definitely more in the skin and in the lymphatic system than in other tissues, but one would think there would be more than just that too. Yeah. So a couple of numbers here. So again, bone marrow is about 80% neutrophils. So it's not really a surprise. And lymphatic compartments are about 85% lymphocytes. And the GI tract has about 70% lymphocytes, and that's a high number of plasma cells, and that's what we would expect. But I was struck by the fact that 25% of the immune cells in the gut are mast cells. That's another cell type that we kind of like.
Why are you shaking your head, Brianne?
Because usually my first thought of mast cell is, man, and now I have allergies.
Yep.
Yeah.
I think of mast cells associated with their pathological functions far more often than I do with physiologic functions. And so a quarter of them being the immune cells in the GI tract makes me, are you sure this person's healthy? Mast cells seem bad.
Oh, really? I just thought, that's a missed opportunity. We have kind of been ignoring the fact that there must be a lot of mast cells in the gut. They're probably doing something important.
Right. Well,
I bet they're sloughing things away.
Maybe.
Because I know we did a parasite paper in TWIP a long time ago where they said the mast cell response helps you slough parasites in certain. So maybe the gut is a big one, because a lot of parasites there.
I think the point that I realized was like, oh right, I shouldn't just think about them in terms of pathology.
Yeah. They do good things and bad things. But we usually focus on the bad. That's human nature.
Mm-hmm.
That's what we complain about with the science reporting all the time, right?
Yep. So coming back to mast cells, I was also surprised to see that 30% of the lung immune cells are mast cells and 30% of the skin immune cells are mast cells. So there's quite a few mast cells in some tissues that I hadn't really contemplated. The other thing I found interesting was 40% of the immune cells in the lung are macrophages. So clearly, I think this is telling us that some of the cells that we know are important, but we haven't really thought about how important are they. The numbers are kind of telling us. And mast cells are probably doing a lot more than we give them credit for because there are a lot of them in some of these tissues.
Right. Okay, so now I want to know, and this is slightly, getting real off topic, but, do we have like mast cell deficient mice? And
Yes.
do we know things about their lung or skin or digestive physiology?
I do not. I'm sure people do. We definitely have mast cell deficient mice because then you can take mast cells from another mouse and put them in. So that's how people have been studying mast cells. But
yeah, because I was thinking if it's this much of a percentage, you'd think that there'd be a decent phenotype when those cells were missing.
I know they're not happy, so I think they're called scruffy mice or something because they're like their fur is all messed up. If I might be mixing that up. It's been a while. But yeah, I would say that somebody should probably go back and look at the original, papers that describe mast cell deficient mice and go back and look at their histologies and things from gut and lung and spleen if they were done. And if they weren't, maybe go back and do them and see, because there's probably a really important role for mast cells in some of these tissues. The next thing that I thought was interesting is the liver is 70% macrophages of the immune cells, right?
Wow.
Of all of the immune cells in the liver, 70% of them are macrophages. So clearly macrophages are doing something important in the liver. The liver filters a lot of things, so macrophages tend to clean up things. So maybe that's a major part of what they do.
So they're sitting around eating.
That's what they like to do.
Yeah.
These are macrophages that are resident there. They don't move in and out, right? I guess.
I don't know that they differentiated between them. They mentioned something later on about, and it's an important point, is there are tissue resident macrophages and there's infiltrating macrophages. And I don't know how much, I can't remember if we've covered that on this before, if I've mentioned it, but the tissue resident macrophages are seeded during development. And then the monocyte-derived macrophages populate the tissues on a replenishing basis. Although the tissue-resident ones we now know can proliferate. We thought they were, terminally differentiated, but that's not in fact true.
Yeah. Well, it would also be interesting to know how much of this is sort of that cleaning up and how much of it is potential surveillance.
Correct. Yeah.
If we're having in an organ that's filtering.
Correct.
Surveillance would be a good thing to do there.
It would be. Yep. So they flip it around and say, okay, so if you look at your favorite cell, where is it? Right? So T cells, B cells, dendritic cells are mostly in the lymphatic system. 70% of all plasma cells in the body are in the GI tract. So that one kind of makes sense, although I was taught that a lot of plasma cells go back to the bone marrow and hang out there and make antibodies. But that's got to be the maximum of 30%, considering some sit in other places too, like in the lung and sitting in the lymph node. The vast majority
you can't
of them
even
go to the
see
gut.
Bone marrow. You can't even see red for the plasma cells.
You can't. Yeah. So let's see. Neutrophils, eosinophils, basophils, monocytes are mostly in the bone marrow. With a little over 10%, around 10% of them in the blood is what I see from that based on my calculation. And mast cells, NK cells, and macrophages seem to be just distributed everywhere. Their numbers can vary differently, but their distribution is sort of everywhere. So the idea here is that lymphocytes mostly sit in the lymphatic system, except for the plasma cells sit in the GI tract and Most of the neutrophils and other granulocytes are in the bone marrow, and then mast cells, NK cells, and macrophages seem to be everywhere. So there's some, if your favorite cell might be disproportionately in one tissue versus another tissue. So as far as total number, so now I also found this very, very, very interesting. So if you count the number of total cells of your favorite cell type, one might say, okay, what do you think are the most abundant immune cells? A lot of people probably say neutrophils, and you're right. So there's approximately, from my calculation off this figure, 650 times 10 to the 9th neutrophils in the body, right? The next best one is T cells, and that's 400 times 10 to the 9th. And then under that is about half that is B cells and macrophages. So about 200 times 10 to the 9th macrophages, 250 times 10 to the 9th B cells, 400 times 10 to the 9th T cells, and 650 times 10 to the 9th neutrophils. All the rest of the cells are super minor in comparison. So I thought that was pretty interesting.
So then that makes you wonder again about those mast cells, because they're in the list of minor in comparison.
Super minor in comparison.
So maybe that's why we know so much less about them in a physiologic sense.
And they're not in the blood, so you can't easily sample them. Right. Yep. So then they got to what I thought was the most mind-blowing part of this whole thing, and that is, how much does a cell weigh? And what is the total weight contribution of each immune cell type? And I just thought, who thinks about this? Okay, so if you're interested, you're ready. If you want to ablate your immune system to try to lose weight, I do not recommend it, because the total mass of your immune system is approximately 1.2 kilograms, which is, a little over 2.2 pounds-ish.
Yeah.
And you would feel really bad.
You'd be in big trouble. So
it's not worth it.
But if there's one population what were you going to say?
You should get rid of other cells, not immune cells, right?
Yeah, I guess the brain cells, right?
Well, the most numerous ones are the red blood cells. You can't get rid of those, right?
That would also be a problem.
Although I don't know if they weigh the most, right?
Probably not, because they're so tiny.
Yeah. I don't know if they make up, the vast majority of the cells in the body. When you put even a teeny weight to them, it's gonna be a significant amount.
Yeah.
I don't know. Okay, so let's talk about weights of organs. This is kind of cool. So your lungs weigh approximately 120 grams. Your blood,
Wait, is this how many grams?
Oh, of immune cells.
Isn't it how many grams of immune cells? Yeah.
Immune cells in that organ. Sorry. You're right. Thank you for correcting me. So lungs have 120 grams of immune cells. The blood has 10 grams of immune cells. The liver has 110 grams of immune cells. So lung and liver are pretty similar, and so is skin at 80 grams. Then the heavy lifters are the lymphatic system. So surprise again, 300 grams, and the bone marrow at 400 grams. Okay, so what, right? So who weighs the most? What cell do you think weighs the most?
Well, I already read it, so.
Yeah. It's a macrophage. They're big on everything, right? And they're bigger by double than any other cell type. And it's an average, because some macrophages are quite small and some are really large. So, they mention that as an issue with this, because they're very variable in size. But a macrophage, in and of itself, I forgot the exact number of an individual macrophage. Did you catch that number?
It's
in supplementary figure 6 or something like that. But total macrophage weight in your body is 600 grams.
Had it even occurred to you before? I mean, no. I know that you also think about macrophages a lot. I have a whole bunch of macrophages in an incubator right now. The thought that they were heavy, did that ever occur to you?
No, they do not weigh down my incubator.
Right.
Oh, I found it. Most cells weigh a few hundred picograms each, and a macrophage weighs a few nanograms each. So the total weight of macrophages in your body is about 600 grams.
Now I want to like go weigh cell pellets.
I know.
And what's the smallest or the lightest?
Well, they only go down. So neutrophils are about 300 grams, so about half the weight of a macrophage.
Hmm.
And T cells, mast cells, and dendritic cells are about 100 grams each. And then they just said the rest are really, really low. I could not,
Yep.
from the thing, tell exactly what they would be. So, what they go on to say, though, is even though some of these are very large cells, there's not that many of them. And then some are smaller cells, but there's a lot of them. And so the total amount of weight in your body doesn't equate to the number of immune cells that you have of that cell type, right? Because we said that the largest number of immune cells in your body are neutrophils by more than double the next highest, which is a T cell. And yet macrophages are double the total weight of neutrophils in the body. So even though macrophages are a third of the size of a neutrophil, they weigh a lot more.
There are a third of as many of them.
There are a third as many macrophages as there are neutrophils. Because neutrophils are so heavy, their total weight is twice as much as the neutrophils. So we come back to like where we started, you divide the cells into lymphoid and myeloid. So approximately 40% of your cells are lymphoid cells. And all together, they make up about 15% of the mass of all immune cells. The myeloid non-granulocytic are about 15%, and then granulocytics are about 43%. And then the granulocytic cells make up a large amount of the total weight because there are so many neutrophils. And then macrophages make up a large amount too. So what is the largest immune organ in the body then, right? Come back to that initial question that we all thought about. We thought, and what the literature says really is the most immune cells are in the gut. And I think what this is telling us based off these calculations is that really the bone marrow and then the lymph nodes or the lymphatic system really are the largest immune organs, but that there's really important distribution of cells in different organs. And so plasma cells, a large number of them are located in the gut. So that makes sense. What I reflected on is, if I know you guys will remember when I think it was when we had Dabrowski Herbert on, we said every cell is an immune cell. So then that changes everything, right? So we're really, when we're talking about all the data we talked about, is these are hematopoietic derived cells that are in the body. But in fact, when we think about what cell can defend itself against infection, really almost every cell is equipped with that capability. So we're saying immune, but it's really circulating hematopoietic derived immune cells.
So the other thing that's interesting is the bone marrow, right?
Yep.
In this aged person, but as you age, the bone marrow goes away. So I wonder if they looked at an 80-year-old, what this would be, right?
So, right. So they did comment on this. They said that the bone marrow is most active right after birth, but it does decline with age. But they said that the total immune mass was relatively stable. So for decades, but that in much later in life, the size of the bone marrow, the cellularity of the bone marrow can decrease by up to 40%, which is a lot. And they said based on the distribution in their calculations, that would be a reduction of about 20% in the total number of immune cells. So that's a significant impact on the immune system with aging. Yeah. And I usually see it related to cell function, but not necessarily related to total immune cell number.
Yeah, I was sort of starting to reflect on something else and tried to do a little bit of back of the envelope math and realized that I needed to like concentrate on the math if I was really going to do it. But I'm starting to think about this number of lymphocytes and what exactly that says about something like a burst size of how much a particular clone of T cells takes over. If we, you could use this number of, say, what is it? 7.6 times 10 to the 11th lymphocytes, 60% of which are T cells, and then start to divide into different clones and think about how much of a change to those numbers you would see when you started to have clonal expansion. And then I realized I needed to actually concentrate if I wanted to do that kind of math. It was too hard.
Yeah, I don't know what that percentage would be, but if I remember correctly, we usually say they expand up to the millions
Right.
for one clone. But what I don't know if you remember this. So
But
many years ago,
if the whole maximum amount is like 10 to the 11th, then expanding to 10 to the 6th is kind of
it's still
amazing.
yeah. It's still
It's
a
like
tiny fraction, right?
Yeah.
I mean, because a couple
I
of
guess that's-
zeros and you're already at
yeah,
like
I guess
0.1%.
I guess it's out, 1 out of 100,000.
Still.
Yeah.
It's still a fair amount. I think what's remarkable to me is that I think it was Mark Davis's group a number of years ago tried to calculate what the representation is of one particular clone of T cells in a naive animal, and it was on the order of 10.
Yeah, I've seen that. I remember that.
So if you think about that number of T cells in the body, now if you sampled an adult human, again, that it's been infected so many different times, it's not comparable. But if you start with that number, let's go back to a child, and the number was estimated to be about, I think it was, I forgot, one times 10 to the 12th total immune cells. If in that child, that for a 10-year-old child of about 70 pounds. So that was the estimate of one times 10 to the 12th immune cells. If you think about a distribution of clones, of naive clones, at a relative ratio of, let's even give it a tenfold, let's say a 100 representation, that's still a fathomable number of clones.
Right.
It's huge. Yeah. So coming back to that, a child, female and male. So we said the male was about 1.8 times 10 to the 12th immune cells. For a female, it's about 1.5 times 10 to the 12th. And for a child, it's about 1 times 10 to the 12th. And one of the things that I was curious about as I was reading this, and they did touch on it, not only does bone marrow reduce across the lifespan, as Vincent mentioned, but one of the things that we always talk about is the thymus involuting, right? And they talked about the number of cells represented and the density and the loss. It turns out that it would be less than 10% of total immune cells that are reduced as the thymus involutes. So it actually ends up being not a huge impact on the immune system.
Not as much as the bone marrow, right?
Right. But it's the type of cell that you're making, right? That's important.
Right.
Yeah,
and I wonder how much about,
right.
if you add in the fact that there ends up being more adipose tissue in the thymus, and those adipose cells are bigger. If like the crowding, the lack of space also influences it.
Yeah.
Probably. I guess I don't know.
Yeah. They also talk about adipose tissue and the fact that macrophages increase significantly in adipose tissue with age, and they're involved in inflammaging and a lot of other metabolic issues that come with increased adiposity in the body as well. But I think one of the big questions is, what about disease states? You were talking about expansion of immune cells. I went and looked, and I found something that if you incubate pet shop mice with SPF mice, so pet shop mice are what we call dirty mice. They have lots of different pathogens and things. And SPF mice are specific pathogen free. It doesn't mean they're totally pathogen free, but they have a very limited number of pathogens. That doesn't mean they don't have microbes, because they have a commensal microbiota. They have a limited number of pathogens. And if you co-house these mice, all of the microbes that pet shop mice have, they can transfer to the SPF mice. And so they looked at cellularity, the overall cellularity in the SPF mice. And they did note that there was an increase, depending on the publication and things, in the liver and the intestine, the kidney, of between 25 and 100% in size and cellularity. So there's certainly major changes in immune cellularity of organs during infection, not just the lymph nodes, right?
Do we think a healthy human is more akin to a pet shop mouse or a SPF mouse?
I would put my money on a pet shop mouse.
Right,
We're pretty dirty.
For sure.
We're constantly not breathing purified air, and yeah, I would say we're pretty dirty.
So in fact, that 25 or 100% increase might be the human baseline that they're talking about here.
Yes. And it's important to think about, though, what experiments do we use for immunology studies? We use SPF mice. So what we're using is not necessarily representative of human. Yeah. I mean, we know, what is it? Mice lie? What's the
mice
thing?
lie, monkeys exaggerate.
Yes, mice lie and monkeys exaggerate. So,
yep.
It's like you knew that Wednesday I'm talking about the pet shop mouse paper in class.
Cool. Yeah. So the last thing I wanted to mention is that also with infection, we already mentioned it with neutrophils, there's also a redistribution of cells, right? So you might find them in different places. And when you sample a healthy human, even though they're healthy human, it doesn't mean they're not exposed to anything, right? Because infection is not equal to disease, right? So what we see in this human, may be generally representative, but it definitely doesn't mean there's no infection or any exposures going on at that particular time point. And again, this is a conglomeration of many different papers to make an estimate. So what was actually going on in an individual, we would probably not know until we have some light beam technology that can enumerate this.
It would be so interesting to look at infections, right?
Yeah.
In different organs and how they skew these numbers, right? Because then you could see cells moving around and get many more in lungs or wherever your infection. That would be very cool. Yeah. But we can't do it now.
No.
Too bad.
And we know that, lymph nodes swell, spleens swell when you get infections. Yeah. So, and that's due in part to increased cell number.
Very cool. I really like this. It's very good.
I'm still stuck on the heavy macrophages.
I know.
Fat Max. That could be a title.
Fat Max. Yeah. Big Mac.
Big Max. Oh, it's so cool.
Yeah. I just, these numbers kind of just blew my mind. But yeah, all of your immune cells together weigh about as much as your liver. So.
That's cool.
Yeah.
Thank you. We have a few minutes. Let's read. We have 2 emails. Why don't we read them? Why don't you take the first one, Brianne, since Cindy's been talking for a long time?
Sure. Rona writes, Dear scientists, 71 was a great episode. I have a question about a widely used drug and how it can influence morbidity from SARS-CoV-2 infection. A family member takes dupilumab injections for eczema. The serum contains IL-4 and IL-13 blockers. In theory, would dupilumab users have a more mild case of COVID? Have studies been done on this in human or mouse models? I do not know of any studies.
I'm not aware. So this is coming back to Vincent, the paper we were talking about where parasites can prevent pandemics. So the data show that, and this is the important thing, right? In the K12 transgenic, ACE2 transgenic mice that you can infect with SARS-CoV-2, if they pre-infected with a parasite, it reduced the disease.
Right. So I think that would suggest to me that if we blocked IL-4 and IL-13 as the letter writer suggests, we would have the opposite effect. We
Hmm.
should be stopping a Th2 response. And so that might give you a worse case because that paper that we talked about implied a Th2 response was protective. And here we're getting rid of the Th2 response with this drug.
Yeah, because I think there was also data in that paper that was suggesting that individuals with allergies or it was asthma, right? That there was some suggestion that those individuals had a milder disease.
Yeah. So I think if anything, this eczema injection would potentially, you would hypothesize perhaps it would be associated with a more severe case.
Yeah.
But no, I don't know of any studies that have tested this particular drug and COVID.
No, I don't either.
Rona continues, I have a separate comment. Perhaps you can invite as a guest Moises Velasquez Manoff. You have heard of him, right? He is a science writer who focuses on health, science, and the environment. He is the author of the book An Epidemic of Absence, a new way of understanding allergies and autoimmune diseases. Thanks, Rona.
You guys heard of this author?
I feel like the title of the book sounds familiar.
Yeah, but I don't know him.
But I have not seen the name of the author.
Neither have I. He wrote an article for the New York Times called The Mystery of My Burning Esophagus.
Huh.
Cindy,
I hope it got better.
Yeah, I do too. Cindy, can you take the next one?
Sure. Oh, one thing I was wondering is, do you think that person's name is Rona, or do you think they did that as a joke?
No, she actually visited the incubator a few weeks ago.
Oh, really?
Yeah, it's
okay.
Rona. She lives out in Long Island. Yeah.
I wonder if she got teased with her name during the pandemic.
Yeah, that's a good question. I didn't ask her. Hey, Rona, you're listening. Did you get teased?
Yeah. Okay, so back on topic. Philip writes, I loved this most recent episode. I worked with NEPO when I was studying ILC2s as an alternative model of activating innate type 2 responses. Cultivation of them involved purchasing stuff from the local garden store, which I always thought was pretty cool. That'd be interesting. I'd like to hear more about that. Anyhow, to your questions about what role type 2 responses could play in modulating other infections like SARS-CoV-2, I would direct you to a bunch of the work that my lab published in the late 2010s. And there's a bunch of different papers. We can put the numbers in the show notes if we want to, but it's one is we found that mice with activated type 2 responses in the lungs were protected against lethal Staph aureus sepsis, and we found that patients with sepsis were less likely to have allergic diseases, maybe suggesting that allergic diseases were protective. They also showed we identified eosinophils and ILC2s as critical in mediating protection against staph sepsis. That's interesting. They found survivors of staph bacteremia had activated Th2 responses. Hospitalized patients with infection who also had allergic diseases were much less likely to die in the hospital.
Ooh, I like that one. Can we go back and talk about my mast cells and my allergies related to that one?
You're better off, I guess.
It sounds like it.
But I think it comes back to the issue of, things that we think are detrimental because they're causing a problem, evolutionarily probably had some beneficial role.
Yeah.
Very true.
So, maybe you have allergies because somehow that type of response was protective in other scenarios like staph bacteremia or something.
Maybe I'm going to have to, give my mast cells a break and not get so mad at them.
So there was one more, activated type 2 responses in the lungs protected against pneumonia lung injury. And I thought this was interesting because, like you, when the pandemic hit, my mom has asthma and my sister have asthma, and they're like, "Oh, are we worse off? Are we going to die?" And I said, "I don't know." But now it seems like maybe those it might actually be protective. And
Mm.
I can't say that I've gone and looked to see whether there are actual studies looking at data, seeing whether
Yeah.
asthmatics are more or less. I mean, there's other issues, right? It's not just a type 2 response. They have more fibrotic lung, they have damaged lungs from having multiple episodes of asthma and repair. So sometimes it's also associated with COPD that comes along with that after a while of damaging the lungs. So anyway, Phil writes, there's a lot of other stuff out there that hints at this. I wrote a whole KO8 around this, testing this hypothesis that type 2 responses are critical to counter regulate sepsis inflammatory responses. It's really fun stuff. I'm glad you're exploring this. Thanks for all you do, Phil. Well, good luck, Phil, with your KO8.
All right, the last one is from Ann. Dear Immune Doctors, I listened with interest to your recent episode about allergies and picky eating. I appreciated the explanations of the allergic response and how it is triggered after a second exposure. I learned this after getting stung by bees several times one summer. While I am fortunate to not have serious allergies or allergic reactions, I do experience the negative effects of mast cells from what I believe is called histamine intolerance. About 10 to 15 years ago, I started to experience pretty annoying rhinitis on certain days for which I could find no explanation. I didn't know what might be triggering the rhinitis. Then, after an evening with friends sharing red wine and cheese, I finally made the connection that these foods were triggering the response. I was miserable for 2 days. Fortunately, this was more annoying than serious in my case. It seems it isn't true for everyone. I've learned to avoid certain foods and take fexofenadine, generic Allegra, when I feel symptoms starting. But I'm curious, especially as I didn't experience symptoms with certain foodstuffs before in my youth. I wonder if more light could be shed on histamine intolerance, which I've seen referred to as a pseudo allergy. It seems that genetics may play a role.
So this was something I hadn't heard about, and I just Googled it. It says histamine intolerance means you have high histamine levels in your body. It can happen if your body cannot break down histamine, and it affects multiple systems in your body, but
Mm.
gastrointestinal systems are the most common.
But it could be age dependent also, as Anne seems to have experienced.
Yeah.
That wouldn't be surprising. Right?
Potentially, yeah.
Cool. All right. Well, that is immune number 73. If you have questions or comments like we have just heard from Rona, Philip, and Anne, you can send them to immune@microbe.tv. And if you enjoy these programs, we'd love your support. You can go to microbe.tv/contribute. It could be cheap. It could be $1 a month. That's nothing, right?
Right.
It's really nothing, and it would help us a lot. Microbe.tv/contribute. Cindy Leifer is at Cornell University. Cindy Leifer on X. I don't know. Do you still use X, Cindy? I don't know.
I do for now. I haven't found
Okay.
a great alternative. Let's poll our audience. If you have great alternatives that seem to be working for you, let us know.
Well, Brienne on Twiv mentioned, what is it, Blue something?
Blue Sky.
Blue Sky. I've been hearing a lot about it.
I'm thinking that Blue Sky seems to work. It's just a little tricky to get an invite to.
Didn't Jolene send us some? Is that correct? You said on
don't
Twitter.
know.
I thought you said on Twiv that Jolene had sent some. That wasn't you?
I don't know.
No? Okay.
Well, send us invites.
Okay.
If you have
If anyone has invites, send them to us. Immune@microbe.tv. I can distribute them. I remember like 15 years ago, my student had invites to Gmail. Nobody could get on Gmail. And she gave me one. I was so thank you so much, Julie. And now, of course, it doesn't matter. Yeah. Brian Barker's at Drew University, BioProf Barker on X. What are you on Blue Sky?
BioProf Barker on all of the social media types.
Okay. Thank you, Brienne.
Thank you. This was a lot of fun. I learned a lot.
I'm Vincent Racaniello. You can find me right here at microbe.tv or on I still am on X because I accumulated 175,000 followers in like 10, 11, 12, 13, 14 years I've been on it. So I'm not going to leave.
Yeah.
Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.